Okay. Now what we will see is we will see cell cycle. Okay, already we have seen in brief about cell cycle. So little bit detail we will see about cell cycle now. So cell cycle, as you all know that in brief I have already told you that cell cycle means the cell will grow and then it will divide, and then again that divided cell can also grow and then divide. So this is just like a cycle. That is why we call it as cell cycle. Okay. So now what we will see is in brief only we will understand cell cycle, because it's not that easy what we people think. Okay. Now cycle means we need to draw it in the circle only. Okay. Some of the things I'll just try to explain you. Okay. Is this a circle? Okay. Clear. Okay, and let us consider this is circle. Okay. Now, what we need to understand here is cell cycle. So we are talking about cell cycle. The thing what is here, we are talking about that. Anyways, I'll write once again. Cell cycle. Okay. Now, cell cycle is divided into many stages and many phases. But I'll try to explain you like in a crude way only, and we will go step by step. First of all, there is a division like this. If this is the center of the circle, it's like this. So what you can see here, or what you can understand here, is this part. First of all, cell cycle is divided into two phases. So by the time I'll be just writing this side also. So cell cycle, first of all, it is divided into two phases, and this phase we will see which are the phases. The first phase we call it as interphase. What do we call it as? We call it as interphase. And the next phase is called M phase. And what is this M phase? I'll tell you. So we will write it here. First phase is interphase. And then what do we have? We have this M phase. So M means mitosis. So mitotic phase or mitosis phase. Okay. So understood now? M means what? M is nothing but mitosis phase. So cell cycle is divided into two phases. The first phase is interphase and the next phase is M phase. Now if you just see this portion, what portion I have given for interphase, then only you can understand that interphase, or it has a long time to take place, or duration of time is more here. Yes. Remember, cell cycle differs for different organisms. Now if you consider human being, the cell cycle, complete cell cycle, to complete one cycle, cell will take 24 hours. How much time it takes? The cell will take 24 hours to complete one complete cycle. That means to grow and synthesize the DNA and again grow and then divide. So this complete cycle, it will take 24 hours. And in that means this complete cycle is taking place in which, how much time? 24 hours. And the example I am talking about, humans, in humans. Now there is another example I'll give you. For example, if you take yeast, yeast is a fungus. Now yeast, if you let that yeast cell divide, it will take 90 minutes. How much? Yeast cell will take 90 minutes for complete cell cycle. Now human cell will take 24 hours. In that 23 hours is interphase, and how much is remaining? Only 1 hour is M phase. 23 hours interphase and only 1 hour it will take for M phase. What is this M phase? M phase is mitotic phase. Now some of the students, they will have this confusions. Why M is mitosis? Why it cannot be meiosis? Some of them have this confusions. So even if you people have this confusion, I'll clear it now only. See, why it is not meiosis? Because as I've already told you, cycle means what? Once the parent cell divides, and the daughter cells also should grow and then divide. So if this continues, this is called cell cycle. Now here if it is meiosis produces what? It produces gametes. And do you think gametes will grow and then gametes will divide and again gamete will produce one more gamete? No, that is not the condition only, no, in meiosis. So meiosis is not considered at all here. No, it's not meiosis, it's always M phase. Mitosis is a division which actually takes place in the form of a cycle. It can be continuous growth, that is why. So this is M is called mitotic phase. I hope this much is clear. Now what I was talking about? The duration of time. 23 hours is interphase and only 1 hour is mitosis phase. Earlier scientist people had thought that interphase is a resting phase wherein cell is not doing anything. But it is proved wrong. Interphase is the stage or the phase where exactly cell is more active compared to this part. It is more active. Metabolically the cell is more active in this interphase more than division phase. Okay, anyways, we understood the timing. What is the timing for interphase? For interphase it is 23 hours if it is example of human cell, and 1 hour it is for mitosis. Actual cell division will happen here. And what is here happening here? See, we are talking about cell cycle. Cell division only is important only, no? Cell division is important, but cell division takes place only with 1 hour. But cell, it prepares for the division for 23 hours. You can relate it with yourself. Now you people are preparing for your exams from so long. So the preparation time is more. But the day of exam, and that too for particular hour of time, you will finish your exam. Actually your exam is important. Writing exam is important. But that will take place within few hours. But the preparation, lot of efforts and lot of time is required for preparation itself. In the same way, you can relate it here. Preparation for the division it takes a lot of time. That is why interphase is having a lot of time and division is having little bit time comparative to this interphase. Okay, this much is clear. Next what we will see, now again these two things are divided into phases, different phases. Okay, that we will see. First we'll take down this much. Okay, next what we will see that now there is, again I told you there are divisions here also, there are different phases. So first let us try to understand the phases of interphase and then we will come to M phase. Okay, now interphase, I'm just going to wrap this, I'm talking about interphase. Okay, so here if we are talking about interphase, I'll just show it here also. Interphase is divided into 3 phases. Okay, so I'll just show you here 1, 2 and 3. 1, 2 and 3. Interphase is divided into 3 phases. Now what are those 3 phases? First phase is called G1 phase. First phase is called G1 phase. Second phase is called S phase. And third phase is called G2 phase. Okay. So we are talking about what? We are talking about interphase. So interphase is divided into how many phases? 3 phases. Now again you people should not get confused. This is just to make you understand. Okay, let us write it here. It is interphase. From where till where to where? From here, from this part to this part. Completely this is interphase. Now in interphase there are 3 phases: G1 phase, S phase and G2 phase. Okay. Now what is this G1 phase? What is this S phase and what is this G2 phase? Now see here, I'll rub this also now. I will be needing more space. I'll rub this also, okay? Hope so the timings you know very well the duration of cell cycle. Okay, now here what is happening in these phases, to understand that is important. Okay, first of all, before this class, if you are getting a little bit confused about this class, already I have done part 1. In that I have already told you what exactly is the cell cycle. So here it is little bit detailed. Now as you know that once the cell is produced, okay, now let us consider there is one cell over here. This is cell. I told you already in that class, in the previous class, part 1, cell cycle, cell division part 1, I have explained you what happens in this phase. This is also called as gap phase, because it is a gap between division phase and synthesis phase. So we will see in detail what it is. So this one, G1, you can also call it as G1, you can also call it as growth phase, or you can also call it as gap phase. It's left to you. But more convenient one is growth phase, because what is happening here is the growth. What growth is happening here? As I have already explained to you in the part 1 of this series, so we have seen that the cytoplasmic content is increasing. What is happening here in this phase? The cytoplasmic content is increasing. Nothing is happening inside the nucleus. Nothing. That is why I have shown empty. And don't think that cell is not having any DNA. No, I have shown this empty to make you understand that in G1 phase, whatever things are happening, everything is happening in the cytoplasm of the cell. So what all is happening here? First thing is the cytoplasmic contents are increasing. Food is prepared. You know, the food is required for the cell to do all these things. Organelles are doubled. Means the number of organelles are increased. Okay. And then other cytoplasmic contents are also increased. Now, what is the next phase? It is yes phase. I told you already. Yes means what? Here, yes का मतलब है synthesis phase. This is very important phase in cell cycle, and that too in interphase, because this is the phase where exactly the DNA is doubling, or the DNA is increasing its content. It is producing its own copy, you can say. Complete amount of DNA, whatever it is producing its own copy. Where? In the yes phase. So, if you consider that, so what is happening here in this yes phase? So here I will show it inside only. Now the cell within the nucleus, already the cytoplasmic content have been increased. Where? In the G1 phase. And then what is happening in the yes phase is the chromatin, or the DNA what is present here is producing its own copy. Like this. Okay. It will not produce the separate copy in a separate sense what I have shown the diagram. I have just trying to explain you in, convince you in your own thinking. That means it is producing another copy of its DNA. Okay, what is happening here? What is synthesis of what? Synthesis of DNA. Synthesis of DNA. Now synthesis of DNA means what exactly is happening here? How it is synthesizing its DNA? To understand this, you need to understand again the chapter from second year, that is molecular basis of inheritance. But anyways, I'll try to just give you some idea about that. So what actually is happening in this phase? I'm showing it here. So in the yes phase, what is this yes phase called? Yes phase is called synthesis phase. And what is happening in the synthesis phase? What is happening here is replication of DNA. What is happening here? What is happening is replication of DNA. Whatever the amount of DNA is present in the cell, the complete amount of DNA will replicate. What do you understand by the term replication? Replication, it's just like Xerox. Okay, if you take one original copy, take Xerox of it, the other copy is similar. Only thing is, you will, if you want, you can take color Xerox or whatever Xerox. That is a different thing. So here what you can understand is, it's just producing its own copy. We will see using a single strand, little amount of DNA. We will see how it happens. So here, if you consider this is DNA, this is a single DNA. This is how the DNA is present here. but it is formed in this condition. That's it. It's DNA only, like this, but it is formed in this condition. We call it as chromatin. Slowly, it is found in this condition only. This is nothing but DNA only. Now, I am just explaining you what is DNA replication here. What happens is, I am just explaining you the crude thing, briefly. So, what happens here is that this DNA will open up. How? It will open up. Both the strands are separated like this, and the rest of the things are again connected here, but only some particular area of the DNA will open. Likewise, complete DNA will get replicated. Huh? Then what will happen? This strand will produce its own another DNA like this, and this strand also will produce another DNA like this. What happened now? This DNA just opened up, and for one strand of DNA, a new DNA has been formed, and on the other strand of the DNA, another new DNA has formed. Do you think now it is, what has happened here? The content of DNA has increased. Yes or no? The content has increased, but it's not increase in chromosome. Okay, I'll just explain that. Don't get confused with all that. So what you try to understand here is DNA will just replicate. So this is the process of replication. Ultimately, if you just see in this form only, what will happen now? You can say that it has formed one DNA, has formed what? Two DNA. Yes or no? Completely. That is why I have shown it like this. Because this one has formed these two. And do you think this is existing now? No. Only these two. And I have just shown you different color to just make you understand how the formation of replication takes place. But the red colored one DNA is also the same thing only, no, which was earlier. So these two DNAs are completely similar. They are similar. So you cannot say these two are different DNA. No, they are similar, completely similar to the parent one. So this is called DNA replication. Clear? So what happened here? In synthesis process or in synthesis phase, DNA replication will take place. Content of DNA is increasing. Clear? Okay. Now, so here, okay, one more thing what you can say. Content and haploid diploid, again it is coming here. I'll be explaining. Already I have explained in the previous one. Again I'll be explaining here, but please watch that previous one and then come here to understand clearly. I hope synthesis phase is clear. What is happening in synthesis phase is the DNA is replicated. Along with DNA, there is one more thing which is happening here. You should remember. Along with the synthesis of DNA, one more step what happens is, here in the cytoplasm, already in the previous chapter we have done about microtubules, centrioles, yes or no? Centrioles will produce spindle fibers. Now what is going to happen in the next is, cell is going to divide. When cell is going to divide, it should form the fibers, spindle fibers. Now for the formation of spindle fibers, what is important? Centrioles are important. Now what should happen actually? I'm explaining you here. So this is cell. Okay. Now already all this in synthesis phase, what is happening is everything is happening here inside the nucleus. Along with that, inside the nucleus DNA replication is taking place. Along with that, what will happen in S phase is the centriole which was present near the nucleus in the centrosome. You know, it was present like this, perpendicular to each other. I told you already, we have done clear detailed explanation on this. This also will divide into two. The centrosome will divide into two and it will form another centrosome. A centriole pair. So what will happen? Another pair of centriole will start moving to the other pole. It has produced another pair which is moving towards another this one. So what is happening here is, why it is produced? Why centrioles are producing another pair of centriole? Because to form spindle fibers. So what you can say here, along with synthesis of DNA, this is first thing. What is the second thing happening? Centrioles or centrosome divides to form centrioles. Centriole pair. Pair means what? centriole is dividing and forming another pair of centriole. So now what will happen now? This pair of centriole will try to move towards this end now, like this, and already this is here like this, and then this one only moved here. Now finally what is happening is nuclear content will be dissolved, okay, like this, and that is in the next phase. Why I am just explaining you what is happening here is this centriole is producing the fiber like this, and these fibers are what? These fibers are spindle fibers. Later on it will produce spindle fiber when here somewhere. But what we are trying to understand, to form the spindle fiber, centriole should divide first only, no? Next is spindle fiber formation is important in the division phase. So in S phase, DNA replication is happening. Along with that, in the cytoplasm, what is happening? Centrioles are dividing. Another pair of centrioles are produced in the cytoplasm. So please, you need to remember that. Clear? Centrioles pair means centrioles divide to form another pair. And where it is happening? It is happening in the cytoplasm. So remember where it is happening. This is happening in the nucleus. Synthesis of DNA in the nucleus. What about centriole? This is happening in the cytoplasm. Which phase? All these things are happening in S phase. And the S phase is a part of interphase. Clear? So this is S phase. Now coming to G2 phase. What will happen in the G2 phase? It is very easy. It's not that difficult for you people to understand here. So in G2 phase, the main thing what happens is, because cell needs to divide here. So this phase will be obviously what? Preparation phase for division. So here all the proteins are synthesized. Microtubules are synthesized. Spindle fibers, slowly the microtubules, spindle fibers are nothing but microtubules, no? So slowly that microtubule formation is happening. That is why protein is important, no? For that microtubule, you know that microtubule is made up of protein. So that protein production and all those things are happening here again in G2 phase. So even this is, it will not take that much time, but here protein synthesis again it is happening. Okay, so overall if you want to understand clearly, interphase is divided into 3 phases. So we will just write here the third phase in interphase is G2 phase. What is happening in G2 phase again? It is called growth phase. What is happening? It's preparing for the division. So the proteins are synthesized here, for which proteins are important for the division process. Okay. So interphase is divided into 3 phases. Which are the 3 phases? G1 phase, S phase and G2 phase. What is happening in G1 phase? Because next phase is DNA replication. All the proteins required for DNA replication is happening here. It's produced here. Along with that, organelles are doubled. Increase in organelles. Cytoplasmic content is increased. All these things are happening in the G1 phase. Clear? What is happening in S phase? Exactly in S phase, if you see, the main process what is happening is DNA replication. So the DNA replicates only during the synthesis phase. And then what will happen? Along with that, at the cytoplasm region, centrioles are doubled. Okay, so this is the function of S phase. I mean, the things what is happening in the S phase. Then coming to G2 phase, because after this phase only the cell is going to divide. So the proteins which is required for the division process are produced over in the G2 phase. Clear? So interphase is divided into 3 phases and these are the 3 phases. Okay? And then here what is again important is just to remember these 3 things. Okay? Then M phase we will do in detail, okay, in the next class. So here what you just understand is we are just going to focus on interphase. Okay? And there is one more thing here, that is after G1. I hope this much is clear first. Take down this much and then I will continue. Once again I am going to clear it that we are not talking about M phase in this class. M phase I will do in detail because again it takes a lot of time. So we are just talking about interphase and understanding interphase properly along with some of the things. Okay. Now, okay. So here, if you try to understand one more thing which is important here. Now, in this interphase region, and you know that it is interphase, so I'll be rubbing this. See what I have done. I will just rub everything because I need some space that side. Okay. Now, what you understood? You understood that this is the cycle, cell cycle. In this cell cycle, mainly it is divided into two phases. One is M phase, which is mitosis phase, and I have also told you why it is mitosis phase, why not it is meiosis. I have told you. And here it is interphase, right? Interphase. Now in this interphase, as again you know that it is divided into 1, 2, 3 phases, and these phases are G1 phase. I will rub this interphase now. So this interphase is divided into G1 phase, S phase, and then G2 phase. And I'm just going to talk about this particular phase which is called as interphase. Okay. Now, what I'm trying to explain is that there is one more phase here in interphase. There is one more phase, that is G0 phase. Somewhere here, it's G0. So G0, or you can also call it as G0, zero phase, or it is also called as, one more name is there, quiescent phase. Quiescent phase, okay. Just check whether the spelling is right or no. Q-U-I-E. I hope it is correct. Okay, just check it out. It's called quiescent phase. Now, what is this G0 phase? Okay, and why it is not included in the cycle? Why it is shown outside the cycle? So we will try to understand this. So this phase actually, this is the resting phase. What I'm talking about? I'm talking about G0 phase, which is also called as a resting phase. Now, what actually happens? I'll tell you. Now, once the cell starts growing, okay, and when it grows in the G1 phase, you know that cytoplasmic content is increased, all the growth, whatever is happening, you know what is happening in G1 phase. Once it has completely completed the G1 phase, okay, then what will happen? It will come to S phase. So once it comes to S phase, then it will go to G2, and then division is mandatory. That means once if the cell, if it is moving through these phases, it cannot come back or it cannot stop, because in G1 phase all the needed things are already there. In S phase, DNA has been replicated. Then here again growth has happened. Then what will happen? You cannot stop that division process. It will divide once it crosses this. Now I'll give you one example. For example, if the cell here in growth phase, it needs to synthesize proteins for the DNA replication and all those things, and if necessary elements are not provided for the cell, for example, then what do you think the cell will do? Now, if the things which is important is not provided for the cell, obviously it will not take the decision of, it will not move to the S phase, because in S phase DNA replication should happen, and what is important for that? Enzymes, proteins, okay, RNA, so many things are important, and if those things are not provided for the cell, obviously it will not go for division. Okay, so what it will do? That time, even though if it is in the G1 phase, it will enter G0 phase. That means it will move out from the cell cycle. Move out in the sense, it will just take rest. You cannot say it is dead. No, it is not dead here. It is alive. It is living. The cell is living here, but what it is doing? It is just resting. It is not growing in size. It's not producing any protein. It's not doing anything here. It is just taking rest. And then once the food is provided to that rested cell, again the cell which is entered Z0 G0, sorry, it can come back to the cell cycle. That means it can enter S phase later on. Once the amount of food, whatever the amount of things was required is given to the cell, is provided to the cell, if it gets all the things which is required, then obviously it will divide. It will enter S phase and then G2 and then it will divide. So this phase is called resting phase. So here what we need to understand, so usually in exam they will be asking you the series of cell cycle. So that you need to remember that it is always G1, yes, G2 and M. Whatever the thing is, they will be asking in this only. You should remember that it is like this. However they mix it up, you should know G1, yes, G2, M. Okay. And what about G0? G0 is somewhere from G1 only. G0 is somewhere from G1. That means after mitosis, it can enter G0. Or within this G1 only, it should enter G0. Once it has crossed G1 phase, then it cannot enter G0. No. So G0 will not come from somewhere G1 and S. No. It has to be somewhere here only in this G1 phase or after the M phase. It will grow for some time and then it will take rest because proteins required for synthesis of DNA is not there, or any other reason it may be. What may be the other reasons why it is entering G0? One reason is what? Because of the available material. It is not getting the materials which is required. So it has entered this resting phase. What may be the another reason? Other reasons as I have already told you, every time all the cells in the body are not dividing. Some cells will take up some other function and they start functioning. And that time they will not divide. Now for example, cells of the body, this cell has been produced because of cell division only, no? Like this. Now this particular cell has taken the function of neuron and it is functioning as a neuron now. Now what is neuron? You know that it is brain cell. Okay? This is neuron. I'm just explaining you. I'm just trying to explain you. Now this cell, if it divides, that means it is in the cell cycle process. But for example, if it has stopped division, it thought it should do some function. Now it has changed itself into the brain cell now. And you know what is the function of neuron? It is functioning as a neuron. Now do you think this neuron will again divide? No. Once the differentiation, this process is called differentiation. So once the differentiation has happened, now already differentiation we have understood, plant physiology we have done, and you know what is differentiation. Their meristem cells will divide and then it will form some other tissue or cell, and that is called differentiation. Likewise, in human beings, for example, this particular cell took the function and formed a neuron cell. That means it has moved out from the cell cycle. So that has entered this G0 phase. That means once it has become neuron, it will, that's what I'm trying to explain you. The cells which will enter G0, it is not mandatory that after getting food or something it will come back. No. If it is differentiated, it will not come back. Once it is differentiated, some cells will not come back. Now some cells are there, even after differentiation, they will again divide. For example, skin cells. Now this particular cell has taken the function of skin. It has become a skin cell. Now something happened to us, and any accident or something, or we fell down something. The upper layer of the skin washed off. We got injury and we need to remove it out, or it is already removed. Then after few days, what will happen? The skin cells which are beneath that will again divide and form new skin cells, no? Already this cell has been converted into skin cell. Okay. Now there are so many cells in the skin cells. All those are produced from these type of cells only. They all are also differentiated only. But that means they have entered G0 phase. They are not into the cell cycle process. Now they are functioning as skin. But once something happened to the skin cells, again new skin cells are produced. again this skin cells which are here, because of some injury or something, again it starts producing new skin cells. And we know this very well. If something happens to our skin, after a few days we will find that new skin coming out. Yes or no? New skin is formed. That means even though this skin cells had gone to this G0 phase, again they came back. Again they started dividing. That means again they have started their cycle. So this is like, what you can understand here is, here this is an example wherein once the cell has entered G0, it got differentiated, it is never coming back again. It's gone. Once the cell is become neuron, it will not again divide. Instead it will die, but it will not divide. But here this is the case where it has entered G0 phase, wherein it is functioning as a skin cell. Again when time is required, when the thing is required, when the function is required, that time again it will divide. It can divide. Throughout the life it will divide. So this is also a G0 phase wherein it can come back to the cycle. So this G0 phase, it will not come back. In here, it has entered G0 wherein it can come back and function again like a cycle. Along with neuron, I can give you another example as RBC cells, red blood cells. So there are stem cells in our body which are continuously dividing. Okay, which are dividing actually. Where exactly mitosis is happening. So here, I hope you understood about G0 phase. Now I told you another example wherein because of food. So because of food in the sense not exactly food, okay, that means enzymes, proteins, all these things. So I'll just write that only. Okay, like enzymes, proteins. If all of these are lacking, then what will happen? Again it will enter G0 phase. Now once it is given, again it can come back to cycle. Clear? So this is about G0 phase. It's very, very important for a cell. It's not like every cell will divide every time. No, it will take rest for some time when there is lack of proteins and enzymes and all those things. Again, when the body provides all these things, again the cell will divide. This is one case. Second case is once the cell has differentiated, that time it is, we call this differentiation is temporary differentiation. This is permanent differentiation. Temporary differentiation, that means temporarily it has differentiated. If it wants, it can divide again. Here permanently it has differentiated. Once differentiation is done, again it is not going to divide. Both of the stages are G0 stages only. Okay, wherein cell is metabolically active. Okay, and what it will do, it's active, but it's alive. But what it's doing, sorry, it's not metabolically active, it's alive, but it is not dividing. It is just quiet. It can differentiate. Even after differentiation, it can come back. Are you understanding this? G0 for the sake of food, proteins and enzymes. G0 temporary differentiation. G0 and permanent differentiation. Clear? I hope I made it clear. And okay, what you need to understand here is, along with this, what is the other name for G0? It is quiescent phase, resting phase. It is just resting for some time, and after the rest it can differentiate and it can move out from the cell cycle for permanent or for temporary basis. Take down this much.